So let's now look at one of the most famous viruses in the world, HIV, the human immunodeficiency virus. First off, HIV is an RNA virus. It has 2 copies of its genome inside a kind of normal looking capsid there. And then it has a couple extra features. This layer on the outside, that's called an envelope. And the envelope is covered in these, well, they look like little purple doorknobs. Those are called glycoproteins. And packaged inside the capsid are these 2 molecules of an enzyme called reverse transcriptase. Let's look at how an HIV virus infects its hosts. So the first thing that's going to happen is the HIV virus is going to attach to its host and get access into the cytoplasm. You'll see here these molecules called Cd4 and Chemokine receptors. Those are markers on the host surface that those glycoproteins have to attach to. The glycoproteins go through a number of changes. They change their shape, which allow the virus to sort of bind with and fuse into the membrane. The envelope is going to sort of fuse with that plasma membrane there and inject the capsid and the genome inside. Once the capsid is inside, it's then going to go into the next phase where the capsid gets sort of uncoated. You're going to see it sort of like fall apart here. Now, those 2 little blue things, those are the RNA molecules. And normally we color code RNA blue, red, but here it's shown in blue. They're single-stranded RNAs. That's the SS you saw there. And that reverse transcriptase, that's going to now make a DNA copy. And again, DNA is shown in red here, a little backwards. But that's a now single-stranded DNA copy of the virus. Enzymes will then replicate that strand to make a double-stranded HIV virus. And one of those strands, again, is identical to the original RNA, except it has DNA nucleotides. So now we have this double-stranded DNA. That's what the DS stands for, is double-stranded. And that is going to go through a nuclear pore into the nucleus. It's going to curl up and sort of make a little ball. And then it's going to incorporate into the chromosome. Once it's in the chromosome, it's now part of the genome of this cell. And that gene, those genes on the virus can be transcribed and translated just like any other gene in our nucleus. So here we see transcription forming messenger RNA molecules. Those are now copies of the gene transcripts. Those mRNAs will go into the ER here. Notice again, the virus is sort of like hijacking the cell. It's using all the cell's machinery to make parts. So those messenger RNA molecules will go to ribosomes on the ER. The ribosome, the little purple guy you see here, it's going to translate those messenger RNAs, just like we would translate any messenger RNA. Except these are going to be viral proteins that are being made here. So we see a bunch of different viral proteins being assembled. So there's translation again, and we're making several viral proteins for the HIV virus now. All right, once those proteins are assembled, they may go through some modification. Again, the virus is going to hijack our Golgi apparatus now and use it for its own purposes. Enzymes will cut those polypeptides, fold them, and we can see what we formed here are 2 of those glycoprotein parts. Those will push out of the Golgi through a vesicle, and many of these will be made. And notice they're actually now sitting on the surface of the host cell. The host cell for HIV is white blood cells, various kinds, macrophages, helper T cells, and so our immune system gets infected with this virus. The viral particles will then push out of our own cell membrane, forming the envelope. So that's where the envelope comes from. It basically hijacks and steals part of our cell membrane, and now the virus is mature. The viral RNA copies will get a little piece. That's a reverse transcriptase being attached to them. And now some of these proteins will fold and assemble a capsid. And now our mature HIV virus is ready to go out and infect new cells. So the whole cycle starts over. The worst thing about the HIV virus and many other RNA viruses is that during this whole process, there are a lot of errors in replication. And unlike DNA, there is no error checking mechanism. So HIV goes through a lot of mutations, and we can't keep up with them. That's what makes it so hard to combat, is it changes very, very frequently. Just when we think we've got a drug that may fight the virus, it mutates, and we have to find a new way to combat the mutated version of the virus.